Our biliary excretion, modeling transporters, modeling of permeability and lysosomal trapping, or modeling our skin permeability or our topical and transdermal formulations. We can use our in vitro data to refine and understand these mechanisms and the parameters that are associated with them. And then we can in turn use these parameters to inform our in vivo modeling to have more confidence in our predictions and for the human and for the preclinical species in vivo. So how can Membrane Plus help you? So first of all, as you may have inferred, it can help you to unlock important information from your measured data. You can use your measured data alongside the mechanistic modeling that you're doing in Membrane Plus to unlock important information and really understand what's going on and what the important processes are. It can help you design better in vitro experiments to capture what you need. You can start to understand, what's going to be the most important mechanisms? What are the most sensitive parameters? What are the things that I need to know the most? And you can design your in vitro experiments to take advantage of this knowledge. And as well, as I said, we want to start to define accurate inputs for our GastroPlus models, our permeability modeling, our PBPK modeling, and so on, so that we have a better understanding of what's happening in the system and what's happening in vivo.
All right, real quick, we're going to pause for a moment and launch a poll. If I can jump in here real quick, Jessica, and let me just launch this poll. So what is your primary use of Membrane Plus? Is it informing GastroPlus modeling, understanding in vitro data, designing in vitro experiments, or other? Give everybody a few seconds to respond. Got the results coming in. A few more seconds. You'll see a few responses. All right, looks like we have all our responses. Let me close the poll. And I'll just share the results real quick here. So it looks like informing GastroPlus modeling is our #1 choice here. All right, go ahead, Jessica.
Thank you, Tanya. And the next question I guess we would ask is, okay, who should be using Membrane Plus? Okay. GastroPlus users, of course, our #1 answer in the poll, looking to define some of these inputs that we're interested in, skin permeability parameters, lysosomal trapping, intestinal permeability, Vmax and Km of enzymes, for example, so that we can start to build accurate in vitro to in vivo extrapolation of the different absorption and clearance mechanisms that may be affecting our system. This is also of use to DMPK researchers if you're focusing on the in vitro kinetics of transporters and metabolism. Again, you can use the mechanistic modeling in combination with your in vitro studies to really understand what's going on and with your Vmax and your Km and the kinetic processes. Okay. And then another group I think is really this of interest too is discovery scientists. When you're very early in the process and you're interested in categorizing your compounds, is this going to be high or low permeability? Is this going to be high or low clearance? Do I want to move forward with this compound or not? This is another place where Membrane Plus can be very useful because you can do so many predictions, based simply on the structure of the compound.
All right, I'm going to jump in here for one more poll question. So what is your primary role? GastroPlus modeler, DMPK in vitro scientist, CRO in vitro scientist, or other? Give everybody a few seconds to respond. Still got some responses coming in. Just another couple seconds. All right, looks like we have responses from everyone. I'll just share these results. So GastroPlus modeler and other. Others are number one. Go ahead, Jessica.
Yep, thank you. For the rest of this webinar, I'll talk about Membrane Plus, the features that we've already had, so some of the basic setup, and some of the information that you'd want to know. I'll talk about what's new in Membrane Plus version 3. And in both cases, I'll be talking about the links that we can make between Membrane Plus and Gastro Plus, and how we can do that. So to start off, just sort of a quick overview of the basic mechanisms that we have for our cell monolayer assays. So this is just a simple look at all of the different processes that we have. Of course, we'll have the apical solution with an unstirred water layer. We'll have the cell monolayer here, okay, with the membrane, of course, surrounding the cytosol. The cells will be resting on a filter, and then beneath the filter, you'll have the basolateral solution. And there's also going to be an unstirred water layer there as well. In the apical and the basolateral solutions, there can be protein binding occurring. Okay, and we can also have loss. This can be sort of non-specific loss. You can have plastic binding to the sides of the assay and so on. In the case of the apical side, this can be open to the air, so there's the possibility of evaporation, and we include that as a potential mechanism. And then, of course, we'll have diffusion between the bulk solution and the unstirred water layer. From the unstirred water layer, there can be transport, I should say, across the membranes, of course, the apical membrane or the basolateral membrane. We can model this as passive transport with different membrane entry and exit diffusion. And of course, we can also have active transport. We can have influx or efflux transporters. Okay, and these processes are happening on both the apical and the basolateral side. Within the cytosol, we can potentially have metabolism happening. Okay, and we can also include, there may also be lysosomal trapping. And so we include a model for the lysosomal trapping as well. We also have the compound that can undergo paracellular diffusion. So instead of going transcellularly through the cells, some compounds will go through the paracellular route and go directly from the apical to the basolateral side. So we include that mechanism. On the basolateral side, again, it's going to be very similar to the apical side. The major difference would be that you have the filter here. So we also incorporate the effect of that filter permeability. Modeling of the sandwich hepatocytes is going to be fairly similar, with the differences, of course, being according to the differences in the assay. So in this case, you have this hepatocytes that are contacting the well plate bottom. So you mainly have that that diffusion and that transport between the basolateral solution and unstirred water layer and the hepatocytes. For the sandwich hepatocytes, we also have the bile pockets that are going to be between the cells. So we also include the passive transport into and out of the bile pockets, and as well as also the bile transporters that can be moving compound influx or in the influx or efflux direction. And this model can also be used for the plated hepatocytes when the bile volume is not considered. The third type of model that we include is the modeling of the suspended hepatocytes. So since these are suspended cells, we don't really have an apical or basolateral side, but of course we still have the similar processes. We'll have the incubation medium where you can have loss, protein binding, evaporation taking place. There'll be a diffusion layer surrounding the cells. You'll have the passive and the active transport mechanisms. And then within the cells, of course, you'll have the binding, metabolism, and the lysosomal trapping. So we're able to take these mechanisms into account with the experimental setting of the suspended hepatocytes. That passive diffusion that's of the membrane entry and exit rates we can predict based on we're using a structure-based model. And this comes from a 44 compound training set I won't go too far into the details in this case, but you can see here kind of the fit between our observed and predicted concentration using this model. Okay. We also have modeling as well to calculate that unstirred water layer thickness. Okay, and that's going to be calculated based on the shaking rate. For all of these different assays, you'll have an experimental setup where you can specify all of these different experimental setup parameters that are going to be important for your modeling. So here, the shaking rate is what you can apply, and you can also see visually what the calculated diffusion layer thickness will be. We also include modeling of the paracellular permeability. We suggest using the Gemin model, and we include the Adson model as well. And that's going to be based on the helicodynamical radius, which we can derive from the structure, as well as default physiological parameters that are based on data reported in the literature. So depending on your cell culture setup, you may need to make some adjustments to the physiological parameters to really reflect your own cell culture. This is the way that we model the lysosomal trapping. So you'll have the cytosol on the cytoplasm on the outside, and with has a pH of 7.4. You'll have the interior of the lysosome where the pH is around 4 or 5. I think by default it'll be 4. And then you'll have the compound diffusing across the lysosomal membrane. Okay. So you'll have these VI values going into the membrane and VO values exiting the membrane, and these are going to correspond to those entry and exits for the overall cell membrane, those entry and exit rates. And of course, again, you'll also have the information, the physiological information that you can update according to your specific cell cultures. We also include models for the protein binding and the compound loss. You can specify no protein binding, of course. You yourself can specify a fraction unbound if you know it or if you want to explore this parameter. And we also include the Hill-Langmuir equation. So you can also use this to calculate your protein binding in your assay. We also include models for the compound loss. So you can have loss due to evaporation. You can have sort of general non-specific loss. And then we also include models for the binding to the plasma. And you can model this as fast binding or slow binding. You can import the structure of your compounds into Membrane Plus, similarly to the way that you do it in GastroPlus, if you have the ADMET Predictor module. If you import them, then you'll get estimates for the physicochemical properties of the compound based on its structure. And you'll also have predictions for the KM and Vmax for 5 of the major CYP enzymes, 1A2, 2D6, 2C9, 2C19, and 3A4. Okay. So if you're modeling the intestinal or the hepatocyte cells, you can use this to get an initial prediction for the metabolism. And in addition to the sort of the graphical outputs that you can get and the text file outputs, we also include the Excel output reporting for all simulation modes. So after you've run your simulation, you can choose to save your output as in an Excel workbook and put it into Excel and do any further analysis that you need to do. In many cases, of course, we want to do in vitro to in vivo extrapolation, and we can do this going from Membrane Plus into GastroPlus. Well, just to give an example case study, we can take the compound propafenone. In this case, we had the structure of the compound. I think in most cases, we weren't able to find the phys-chem properties in the literature, but we're able to use ADMET predictor to predict the solubility, pKa, log P, and so on of the compound. This compound is going to be a CYP2D9 substrate, and we know that it has saturable dose-dependent kinetics. Okay. In this case, we also had some observed IV PK data. So we're able to perform an in vivo, in vitro extrapolation using our in vitro data with suspended hepatocytes to figure out the Vmax and the Km, and then use these estimated values as inputs for our in vivo simulation in GastroPlus. So over here, I had summarized the information from the in vitro assay. With suspended hepatocytes, and we know the rate of the disappearance, and also the experimental setup. We can use Membrane Plus to do a fit to the in vitro data, and I believe this is across 4 different dose levels. And so we're able to optimize the CYP2D6 kinetics, Vmax and Km, okay, for propranolone across these 4 different dose levels. Okay. Something interesting that you see in this case is that this optimized Km based on the simulation is actually lower than the value that's reported in the paper. And the reason for this is because the fitted value that we're using here, that we've estimated here, is actually going to be indicative of the true intracellular Km. So this is a place where modeling and simulation can really shine, because you can really start to get at, kind of the true Km values instead of having to use workarounds and make assumptions based on your experiments, and which you're able to experimentally measure. In turn, we're able to use that in vitro Vmax and Km that we optimized in Membrane Plus and convert that into an in vivo Vmax and Km that we can use in GastroPlus. So in GastroPlus, we're able to extrapolate from in vitro to in vivo values, okay? And we can use those in vitro values that we fitted using Membrane Plus. We can use this in combination with the PBPK model of the compound using default parameters and default distribution. And you can see here that using this approach, we can get a very good match to the experimental data. Okay, so in this case, we wouldn't be actually, this would be a prediction, and you can see that it's going to be very close to the experimental data that we actually observe based on the predictive properties of propranolone. And based on our in vitro data that we use to fit the Vmax and Km for the clearance. So what's new in Membrane Plus version 3? What the major aspect that we've added is starting to model the permeation through ex vivo skin using our transdermal compartmental absorption and transit model. So if you have your in vitro penetration test, your in vitro release tests, you can model those using the Membrane Plus program. And you can use these to inform your predictions of the absorption and the distribution in vivo. So this is an overview of the mechanisms that we have in the TCAT model. You can use several different types of skin samples. And in this case, I'm showing the full thickness skin, where we have all of the different sections. Okay. So first, you'll have some formulation that's applied to the surface of the skin sample, I should say. You can have evaporation. This can be evaporation of the formulation itself, or evaporation of the API within the formulation if it's volatile. You can potentially have dissolution and precipitation processes occurring within the formulation. And we're also able to model emulsion dosage forms. So if you have a dispersed phase and a continuous phase, we can also model the release between the dispersed and the continuous phase, and also any direct diffusion from the dispersed phase into the skin. There'll of course be the diffusive processes as we go from the formulation into the skin and through the different skin layers. And the different skin layers can have different permeability associated with them. So a specific diffusivity and specific partition coefficient. Within the skin layers, we could potentially have binding occurring. Okay. If your skin sample is still metabolically active, you can also specify metabolic clearance in the living skin layers, viable epidermis, dermis, subcutaneous tissue. And we also include an option to include sebum. I think there's some question about how much it may be contributing for the excised skin, but we include it as an option if you want to see what effect it has or if you think that it's contributing for your sample. Once you've once you've diffused through the entire skin, there'll be a receiver compartment. So then you can have diffusion down into this receiver compartment. You can potentially have non-specific binding within this receiver compartment. Not sure if I put the diagram there. And if it's a flow-through diffusion cell, then you can also have convective transport into a reservoir. So this is just an overall look at the mechanisms that we include in our TCAT model in Membrane Plus. So just to summarize some of the major features, we want to model diffusion through these skin samples and static and flow-through diffusion cells, such as Franz diffusion cells. We include the modeling of complex topical formulations. So for example, solutions, suspensions, patches, ointments, pastes, lotions, gels, and creams. And again, we are particularly including those emulsion dosage forms as well. We have a few different types of skin samples that can be modeled. And we also include an option for a simple membrane. So either you can use this for your in vitro release testing, or you can use it as a very simple model of the skin where you just have a single compartment. And of course, as I mentioned, we include the processes of evaporation, dissolution and precipitation, the absorption into the skin, the diffusion and partitioning into the skin, and then into the receiver fluid. And we can use, just as we do with the other cell assays, we can use these model parameters to predict the in vivo absorption and distribution in skin in GastroPlus by being able to refine, validate, and optimize the skin permeability parameters and the formulation parameters. So this is just a few slides just to show where you can actually make these types of modifications in the program. You can specify a static or flow-through dermal diffusion cell. Okay. And you can describe the properties of the experimental setup. So the volume of your receiver compartment, the flow rate, and so on. And you can save and reuse these experimental setups. And you can also use this for the other assays as well. And you can reuse these for different compounds and different databases. In the TCAT model, in Membrane Plus version 3, so you want to specify the species that the skin sample came from, as well as the region. We have several inbuilt physiologies. We include the regional physiologies for human and mini pig, and we have sort of general whole body skin physiologies for the rat and the mouse. And of course, you can also create your own custom physiologies as well. We also include the case of a simple membrane, so you can specify a synthetic membrane. There's several different types of dermal samples that you can model. So in the screenshot here, you can see this would be dermatome skin. You can also model the heat-separated epidermis, the full thickness skin that would be including some subcutaneous tissue at the bottom. You can model stratum corneum alone. And again, you also have this model of just a simple membrane. And of course, you can specify as well the skin pH, the total sample thickness, and so on. In addition to specifying the type of the formulation applied to the skin, of course, you would also have to give the information such as the dose that you're applying, the applied surface area, so this would be the surface area of the skin sample, excuse me, the formulation volume and mass, and so on. You also want to you can also specify the formulation properties that are going to be affecting the absorption. So for example, you can specify the solubility of your API in your formulation, which of course may be very different than its aqueous solubility. The diffusivity and the partitioning in your vehicle. Okay. and other aspects of your formulation. As I mentioned, we also include modeling of the evaporation. And when we're modeling the evaporation of the vehicle, this can be modeled as a linear or a nonlinear process. If modeling the linear evaporation can be done using several published equations. Okay. I think our general recommendation is for the Nielsen model, but we include several other ones. And when you select one of these models, you can see the predicted evaporation rate based on the properties of your formulation, and also the time that it'll take to evaporate. We also allow the user to specify a nonlinear evaporation rate. So if you have observed your formulation and you know the rate of the evaporation, oftentimes this will be a nonlinear process. And so you can specify that as a tabulated function that can also be fit to a smooth Weibull function. And in these cases, you would be specifying the extent of the evaporation over time in terms of the percentage of the total volume. If you're modeling emulsions, lotions and gels and creams, you also need to want to give the information about your dispersed phase. Your dispersed phase volume fraction. You can specify the mean droplet radius and specify a standard deviation to have a distribution. Okay. The contact angle of the droplets on the skin, and also the partitioning between the dispersed phase and water. We have two different release models included for the emulsions. The equilibrium model assumes that there's continuous and dispersed phase concentrations are always in equilibrium according to this partition coefficient. And the diffusional model is used for the case where you have a thin membrane or surfactant layer that's surrounding your dispersed phase droplets and stabilizing them. In that case, you can give some values for the diffusivity through that membrane and the thickness. If you have an emulsion that's dosed that has suspended particles in the continuous phase at the time that it's dosed, we also give you the option to include this as well, which is something that you see for some low solubility compounds. If you have some information about your droplet size distribution, you can specify this in a PSD file, and you can do fitting to get the mean and the standard deviation based on any data that you have. And you can do this as well for the suspended API particles. We'll talk in the next slide about dissolution and precipitation. And then just as with the suspended particles, this can then be exported to the Membrane Plus TCAP model. Or it can be used directly from the file. The dissolution processes, if you have dissolution and precipitation occurring in your formulation, these can be modeled using some standard equations, Johnson, Wing-Flanagan, or you can specify a Z factor or assume instant dissolution. As with the dispersed phase droplets, you can specify mean particle radius or particle distribution for these undissolved particles. And you can also specify information about the diffusion layer thickness. And we also include the modification for temperature if you have that information. We model the precipitation as a first order process where you specify a mean precipitation time, and you can specify the form of the precipitate. We model the permeability in the skin layers as well as the formulation using a diffusivity and a partition coefficient. And then these together, along with the thickness, can be used to calculate the permeability of each skin layer. We provide several published equations to estimate the permeability in the different skin layers. So 3 for the stratum corneum, 3 for the viable epidermis and the dermis, and then we have 2 for the sebum as well. And for the subcutaneous tissue, we have an estimate of the partition coefficient, and we have a default value that we use for the diffusivity. All of these can also be user defined, and we do find in many cases that these values need to be refined, and the values from the equations don't necessarily work that well. So this is another place where you can really use Membrane Plus to really refine and understand the permeability, and start to explore these diffusivities and partition coefficients, and potentially do optimization as well. After you've run a simulation with a TCAT model, the simulation tab will show the quick outputs of interest. It does this for the other assays as well. But of course, for these skin samples, the quick outputs that we're interested in are going to be a bit different. You'll be able to see the maximal flux through the skin at the time at which this maximal flux was reached. You can see the cumulative amount in the receiver compartment and the percentage of the dose that was absorbed into the skin sample at all. And then for each of the skin compartments, you'll be able to see the Cmax, the Tmax, and the AUC, okay, for the type of skin sample that you've chosen. So for your model outputs, you can view the time profiles, of course, for the concentration and the amount in the formulation and the skin layers. Okay. So this is giving an example for caffeine, which I can also show within the program, where we can compare the observed to the predicted mass in the vehicle, stratum corneum, viable epidermis, dermis, and also the amount that's getting into the reservoir. You can also look at the flux through the skin and the way that that's changing over time as well, to get an idea of how that's going in your model. And then the final link that you can use is once you've validated your model with Membrane Plus and you understand what's happening in vitro, then you can use these validated parameters and get your GastroPlus simulations to start to predict the in vivo absorption and the distribution into the skin. The TCAT model in GastroPlus is set up very similarly to the one in Membrane Plus. So for example, if you have an optimized diffusivity, partition coefficient in the stratum corneum, for example, you can, once you understand that parameter and you fitted it or validated it in vitro in your Membrane Plus simulations, then you can start to use it in your GastroPlus simulations, and you can start to make predictions about the local skin concentrations in vivo, as well as your plasma concentration profile in vivo. So I won't go into too much detail, but I'll go ahead and open up the program and maybe show a little bit of this. This is showing the example of caffeine in phosphate buffered solution. Okay. For the TCAT simulations, we primarily be looking at the log P, pKa's, solubility, and that initial dose. But for this model, we don't use the passive transport model that we're using for the cell monolayers or for the hepatocyte assays. We can specify our experimental setup, and in this case, I needed to make a model for my particular setup, which had for the receiver compartment volume. And this was in a flow-through diffusion cell, so I'd also specify that. Okay. If your donor compartment is going to be covered, then you can specify that as well. And you can click the button to pop up your transdermal model. In this case, I knew this was with human abdominal skin, and I knew the sample thickness. Yeah. And in this case, the caffeine was applied in phosphate buffered saline. So I can model this as water. So I can use the diffusivity and partition coefficient for water. I include my information about my dosing. And then I would also include the information about the molecular weight and the solubility according to what it is in water. I mean, if I had a different vehicle, then I would adjust all of these to take that vehicle into account. You can double click your compartments as well to pop up the different aspects that are going to be related to it. In this case, if you have the software, you can go through this as a tutorial example. we tested some of the different published equations for caffeine, and they got us in the right ballpark, but we thought that there could be further improvements. So in this case, we actually did some optimization of the diffusivity and the partitioning in the stratum corneum and in the dermis based on our in vitro data. And now we're able to use those values directly in the simulation. So once you've set up your simulation, of course you want to run your simulation results. Okay. And again, once you've run your simulation, you can see the quick outputs that we spoke about. So the maximal flux through the skin, percent absorbed, amount in receiver compartment, and I can see my Cmax, Tmax, and AUCs for the individual skin compartments. On the graph tab, by default, I can see the concentration in the receiver compartment. And I can also look at what the amounts were in the vehicle, in the different skin layers, in receiver compartment, and in the reservoir at the end of my simulation. And of course, I can also go to the new plot window here. Okay. And this is where I can go, for example, if I want to look at the amounts in my different compartments, for example. And in this case, I had experimental data, so I can do a comparison to that experimental data as well. If I want to use these parameters in GastroPlus, I am able to do that, and you can just sort of transfer them over. Okay. So, for example, I might have my similar simulation set up. I want to predict what's going to be happening in vivo. Yeah. Then, in that case, I can open up my TCAT model in GastroPlus. Okay, and I can use the same settings for the skin permeability parameters. So for example, here I can use that diffusivity and partitioning that I optimize using Membrane Plus. I can use this directly in GastroPlus. And then here I'm also taking into account, for example, the perfusion into the systemic circulation, the potential of the sebum route, and so on. And then, of course, I can run my simulation and I can have my predictions, given this formulation and given this dosing of what I expect to get in vivo. Fraction absorbed, bioavailability, plasma concentration, and also, of course, the local concentrations and amounts in the specific skin compartments. So just to conclude, give a general overview. In Membrane Plus, we're able to offer mechanistic models for oral permeability, skin permeability, and hepatocyte assays. We're able to include the passive and the active drug transport in the various assays. We're able to look at the interplay between the metabolism and these different drug transport processes, because of course these will be affecting that intracellular concentration, which then affects the metabolism. We're able to model the drug disposition in the bile for these sandwich hepatocytes. We're able to estimate the drug concentrations within the cellular structures, or in the case of the skin, within the different skin layers. In particular, we're also able to understand effects such as the lysosomal traffic. Using Membrane Plus, as we mentioned before, you're able to unlock important information from your measured data. You can use it to design better in vitro experiments to capture what you need and really understand the most sensitive parameters and most sensitive mechanisms. And of course, you can also use this to define accurate inputs for GastroPlus, whether these be the metabolism, the permeability, intestinal permeability, skin permeability, and so on. So you can use these for your permeability and for your PBPK models. So thank you all very much for your time and attention. Please feel free to contact us if you'd like more information about Membrane Plus, or if you have any interest in our University Plus program, or if you'd like to know what our University Plus program is, please feel free to visit our website. Thank you.
Jessica, you knocked it out of the park. Thank you very much for that excellent and insightful presentation and demo. And we'll now open it up for the question answer session. And there's already been a flurry of questions coming in, so let's get to it. Just as a reminder, if you would like the opportunity to have your question answered live now, please use the questions pane in the GoToWebinar panel. There is also an opportunity to ask your question orally by using the hand raising icon that can be found next to your name as well in the GoToWebinar panel. To kick off the questions, let me invite Mr. Neil Miller, our Vice President of Simulation Sciences, who has been patiently waiting to contribute to today's webinar. A question that came in early as soon as Jessica started talking about our abilities to simulate for different in vitro systems. Neil, what in your opinion is the most valid in vitro system or assessment for predicting or understanding permeability and or bioavailability?
Thank you, John. Can you hear me okay?
Can, yes.
Okay. I think that depends on the company. In my experience, the company that I previously worked for preferred the MDCK assay. But speaking to colleagues who were attending when we could attend lots of seminars and meetings and workshops, it seemed that other companies were preferring CACO2. So I think it's dependent on which company you work at as to which is the favoured assay that's employed for the predictions.
Very diplomatic answer, Neil. Thank you very much. This question is a little bit more on the technical side with Membrane Plus. So this may be directed more towards Jessica. Jessica, for plastic binding in some of the models that we have, is that typically modeled as a saturable phenomena or partitioning approach? Can you go into a little bit more detail?
Not sure if I can go into a ton more, that much more detail off the top of my head, but I believe we do actually include the option to include either type of process. So you can model it as sort of a fast saturable binding. And I believe the slower infinite approach does use more of the partitioning. So we really sort of give you the option of which way you want to model the plastic binding.
Yeah, very good. If I could add a little bit more to that, one of the things that is pretty unique about the Membrane Plus platform is the flexibility which is allowed. And I think it serves as a really nice data analysis tool, which means that you have that opportunity to take some of your data that is being captured in a different in vitro systems and use that to start to better understand and appreciate the impact or influence of individual mechanisms or processes to help explain that data that's been measured. And so in the case of something like binding to plastic, we would be able to leverage some of the data that we have measured, use that to figure out whether or not it is a saturable phenomena or something else which is describing potentially some of that loss. So the flexibility piece is something that I think is somewhat underrated here with the Membrane Plus platform. Another question, I'm not sure who to direct this one to, so I'll encourage both of you to contribute. When it comes to the hepatocyte assays and more specifically the sandwich cultured hepatocytes, should there be both basolateral and apical sides included in the models? Is that something that is considered in the structure?
I'll take a first pass at that while Jessica goes through the slides there. I think as with all of our products, we do provide tutorials to help users get familiar with the software and also explore the power of the simulations. So there is a tutorial for the sandwich cultured hepatocytes, which does include parameterizing the basolateral influx and efflux, plus the bile canicular efflux as well. So that would be my answer to that question.
Yeah, yeah, I would mention as well. I think I may have misspoken a bit before. We do include the apical membrane in the case of the sandwich hepatocytes. But based on the setup, it's just sort of in a different location than with the with CACO2 and so on. So. But as Neil said, I would encourage you to take a look in the software itself and go through the examples that we have for the sandwich hepatocytes to really have a good understanding of how the model is structured.
Thank you, Neil, and thank you, Jessica, for clarifying. Jessica, you teased, and again, a question I think for both of you here. Jessica, you teased during the presentation that especially with the permeability assays built into the software like CACO2 especially, that there's an opportunity to model transporter interactions or effects. Could you comment a little bit more about what would be some of the advantages of using Membrane Plus to analyze some of the data that may be measured across concentrations in some of these in vitro permeability systems to help better inform maybe some of the transporter inputs for GastroPlus models?
Sure, that's a great question. Previously, when I spoke about the case study, we spoke about the case where let's see if we can find it here. Where we're looking at in this case at enzymatic kinetics, and we're optimizing that Km value to indicate the true intracellular Km. I would say being able to do this type of optimization is going to be even more important for the transporters, especially when you're looking at transporter kinetics. Even when you do have the in vitro data and you have the in vitro system, if you have an efflux transporter, measuring that Km directly is not really possible because the true Km is going to be based on that intracellular concentration, and that's not something that you can measure directly. So you can use a similar approach to this enzymatic approach to fit your Vmax and your Km for your transporters, and you can really get a better idea. And of course, using your in vitro information to inform and to fit the model. But in these cases, this gives you more confidence in the Km that you optimize because in this case, you really do have a simulation of that intracellular concentration. And so so the values that you optimize are going to be more indicative of that true intracellular Km. I would say for the influx transporters, it's similar, just as with the enzymes here. But I think especially for those efflux transporters, it's very, very useful to have this in silico approach to help to alongside your in vitro data to really inform your Vmax and your Km for your transporters.
Excellent. Thank you, Jessica. Switching gears a little bit and talking about the new dermal models in the software. Can you comment a little bit on the impact of the type of skin assay that might be considered when trying to measure dermal penetration or absorption? For example, the full skin versus heat separated versus, stripping stratum corneum only.
So I would say it's really going to depend a lot on your compound and on which layers of the skin are going to be most important. So, just in a physical sense, of course, we do need to know, well, what type of skin do you have and what's the sample thickness, just so that we can understand the overall diffusion process. Beyond that, in general, stratum corneum is going to be the major barrier of the skin, of course. We're going to be most interested in the permeability through the stratum corneum. But what we have found is that doesn't necessarily mean that we can just discard what's happening in the other skin layers. The permeability in the viable epidermis and the dermis and even subcutaneous tissue, these can also be affecting how much compound you get that's reaching the receiver compartment or reaching the systemic circulation. And because these also would change the concentrations, they can actually also have an effect on the overall absorption in some cases. So one aspect of it is just, you know, correctly modeling your experimental setup. But the other aspect is there are cases, you know, where we really are interested in the permeability in these deeper skin layers, because this really can be affecting the disposition of the compound. Yeah, I would say, of course, you know, if you can only look at one thing, you want to look at stratum corneum. But viable epidermis, dermis, subcutaneous tissue, and sebum can potentially also be affecting.
Very good. Again, staying with the dermal models, could you comment on any experience that you've had in terms of being able to utilize in silico inputs to predict parameters like diffusivity and partition coefficients for GastroPlus simulations through the skin versus leveraging some of the in vitro data and optimizing some of those parameters? Are there major differences between some of those in silico methods versus what is ultimately fitted, or is it highly dependent upon the type of compound you're working on and so forth?
I would say it really depends on the type of compounds. And of course, you know, we're working towards the case, you know, we can start to give, you know, specific recommendations based on compound properties, but I'm not sure if we're quite there yet. In terms speaking about the predictive equations that we have for the skin permeability, we found that, you know, across a wide variety of compounds, Wayne-Casting-Niche and Kratzos equations are the best when you're across a wide variety of compounds. So if you're just sort of doing screening, maybe you're in that discovery phase, then we would suggest these options. But unfortunately, we still run into these compounds, and of course, this compound, I think caffeine is one of them, where these predictive equations don't necessarily give us the best result. If you do this tutorial, I think you'll see with the defaults, these get us in the right ballpark for caffeine. You know, they're not terrible as a prediction, but oftentimes, you know, if you really want to be sure about it, you really would want to make your own adjustments based on your experimental data. I'm not sure if this is entirely answering the question. When you're moving from in vitro to the in vivo simulation, one thing to think about would be the effect of the formulation. Are you using the same formulation? And if it's different, then there could be some different things going on. You may also want to make some adjustments going from the in vitro to the in vivo for the stratum corneum in particular. The Wayne-Casting-Niche model includes an adjustment depending on the hydration of the skin. And so even if you're using your own parameters, you may want to sort of use a similar offset when you're going from the in vitro to the in vivo case. Currently, we're working on making a firm recommendation on that, but that may be something to think about. When you're looking at the deeper skin layers, as far as we can tell, we think sort of as a default that these would be pretty similar. And of course, the other aspect that you might want to think about is if you do have a compound that has significant absorption through the sebum, you know, could the sebum be contributing in my in vitro case? But we always do expect it to be contributing in the in vivo case. So you would also want to be thinking about those aspects.
We are very fortunate to be able to tap into this wealth of knowledge you have, Jessica, considering the collaborations we've had with FDA and the consortium in Europe. And so everybody on this call is benefiting greatly. One last question on the dermal model. Any experience predicting the penetration in damaged skin or skin that would be non-healthy in some way, shape, or form?
That's a good question. I don't think that we've worked on this in a case that we can share. Speaking about this generally, you'd expect the disrupted skin, particularly the stratum corneum, to have higher permeability than the intact skin. So whether that takes the form of increasing diffusivity or often increasing the partition coefficient or moving it closer to 1, so similarly to water. If you want to model a case where perhaps the skin has been damaged and the stratum corneum has been removed, we can't sort of physically remove it, but if you set the values for diffusivity and partitioning to be the same for stratum corneum and viable epidermis, this would sort of mimic the case where you don't have stratum corneum and compound is going directly into viable epidermis. So I would say, you would want to think as well. So you would want to think about, do I have a damaged stratum corneum? Do I have no stratum corneum? What exactly is physically going on with the system? And then use that to inform your modeling.
Very good. A little birdie is telling me that we're running out of time. So last question, and for those that we won't be able to get to today, we promise to respond via our Membrane Plus and or Simulations Plus LinkedIn pages. But last question for both you, Jessica, and Neil. I'm going to ask you to pull out your crystal balls. And I'm going to ask you to look there and predict, what does the future hold for Membrane Plus? And if I guide you in a certain direction, have there been any considerations to expand the software to try and better capture and model in vitro DDIs? But I'd like to hear both your thoughts on extending Membrane Plus to cover DDIs, and then also any other areas you think we should potentially attack here in the future with additional releases.
I'll start it off, John, in terms of what does the future hold. The future holds what our users require. So what our users need and want is what we would look to include in all of our software platforms. So if that is something that people desire, then we would look to get that in. I mean, what we do with GastroPlus, we have the pharma user group, as you know, and we obviously canvass them for opinions of what's required. So what I would do is invite people on this call to let us know what they need to make the tool even more useful for your day-to-day applications. So specifically, I haven't been involved recently in specific discussions about what's in the next version of Membrane Plus. But certainly if we could hear from the audience today, that would really make sure that we're getting it right for them to help them in their day-to-day work.
Thank you, Prime Minister.
You taught me well, Obi-Wan.
Jessica, do you have any thoughts? I think Neil's answer, I think pretty much covers it. What we want to add things that our users are looking for and that our users want to see. One thing that I would mention that I think is on our radar for the dermal module is also taking into account the variability in the skin samples. We're not able to do that currently, but I think that there would be a lot of interest in this, and really understanding, how the variability in the skin samples and the physiology would also be affecting the results. I think there's a lot of interest in that from the regulatory perspective. But of course, we want to primarily be guided by our users and what our users are interested in.
Very good. Well, Jessica and Neil, thank you guys very much. This has been very fun for me and highly educational. Tanya, can I turn it back to you to take us home?
Absolutely. Again, thank you, Jessica, Neil, and John for hosting a fabulous live Q&A. I think there were a couple questions we may not have gotten to, so as John said, we will post them on our LinkedIn and our Membrane Plus showcase page. I also put in the chat some information about our Facebook and Twitter, if you'd like to follow us there. And I included some links that Jessica mentioned there on her last slide. If you want to learn more about Membrane Plus or our other products and services, please visit our website. And there's also a link there for the University Plus program for anybody who's interested. So this webinar has been recorded for playback, and it will be available on our website and YouTube channel. This concludes our webinar for today. Thank you for joining us.
Thank you, guys. Take care.